Right. So what you saw today is what I described yesterday. That's the data acquisition software. So that's the LabVIEW program that communicates with the National Instruments data acquisition card, which is connected to the TIA, which is connected to your device. So that's how you're bringing in data, including spikes or events that you saw being collected in real time today. So like I described yesterday, our analysis philosophy is to just write everything we collect to a file and then analyze it after the data has been collected. So what I'm going to talk about today is the software that we've written to analyze these events or these peaks, and we just call it Event Finder. And it's continuing to be developed. So there's iterations of it that are being used in the lab. Some of what you'll see today is a slightly older version, but the analysis philosophy hasn't really changed. We keep refining and improving it. But what I'll describe today is basically what we do. And like Dr. Hall mentioned in his presentation, a lot of the way that we analyze the data hasn't changed that much from the 50s when Coulter published his paper. You know, we still look for amplitude thresholds to find events, and then we go from there. So generally, when we analyze data that's collected, we use two different files. So what students have normally done is first collected a background file, which is indicative of the signal that you see when you just have a buffer in your chip. So there's no analyte to analyze at that point. It's just the background. And the reason that we collect that is because we need to have some idea of the overall noise of the system, which then helps us establish an amplitude threshold. So when we have an experiment with events in it that are creating these peaks, we know how to find the peaks. The peaks should be at some level above the noise, but we need a more quantitative way of just saying, okay, we can see them. We have to come up with some way of establishing a threshold to just effectively find them for further analysis. So then the event file is what contains those events, and we apply a lot of the same processing steps to the event file as the background. But what we end up with after all of this is we find the events, we make measurements of the events, and then we'll report those measurements to a file that you can then analyze further in Excel or other software. So I'll just walk through the steps to show you how we do it in the software that we've written. So this is the event finder. This is an earlier version of it than what's being used in the lab now. But this is just custom written software in MATLAB using their GUI design, which they call, it's just their app environment. So you can write basically an app that you can distribute and install as an app in MATLAB and then run. Okay, so what I'm showing here is a background file. This is the raw data that was collected. And what you should see is that on the y-axis, the amplitudes match what you saw during the acquisition in the LabVIEW program. So if this is off by a considerable amount, that may mean that the file gain value here is not correct. And I talked about this yesterday. This is the gain of the transimpedance amplifier. So here it's set up to be 100 nanoamps per volt. So when the data is scaled and displayed, that this axis is represented correctly. Okay, so this is just a background file that was collected. It's 60 seconds long. Here, there's some power spectra and autocorrelation plots, but what's important is just to look at the shape of this. So what we're really interested in is the noise in this data. So basically the extent in the Y direction to establish this threshold that we'll use then to find events in the next file, which is what contains the events. All right, so the first step is to correct the baseline. So you can see before you do that, the data is fairly flat, somewhere around, 9.95, is that times 10 to the minus 8 amps. But to perform this calculation to quantify the noise, it's easier to just essentially remove the baseline. So this is the data after the baseline has been removed. So now you see it's centered around 0 and it's fairly flat. The whole idea being is that you calculate the standard deviation of the background, and then you use that standard deviation multiplied by some factor to give you the amplitude threshold that you'll use to call a peak an event. Yes.
Baseline graph is basically make it equal to 0. So subtract basically negative value of the what you're seeing.
So I think of it as removing a DC offset. So if you have a signal that's basically like, here's your graph, here's 0, but it's riding on, say, like 10 above that, it's removing that 10 offset. So it's now centered around 0. That's what you were saying. Yeah. Yeah. So the way that it's implemented in the software is we apply a high pass filter. If you remember what Colin presented yesterday, what that means is that you get rid of all of the low frequency content of the signal and just preserve the high frequency content. And when I talk about low frequency, I'm talking about really low frequencies like a hertz or below. So a hertz is 1 cycle per second. So if you have data over a window of a second, 1 hertz is just one sinusoid over that whole 1 second period. So it's really low. And below that is even lower. So like 0 hertz is representative of just a DC shift. So to get rid of the stuff that we see here, that's both the DC shift, so just up and up away from zero, but also this very slow trend that occurs over 60 seconds, you can get rid of it by applying, and in this case, it was only a 1 hertz high pass filter that just got rid of that offset. Yeah. So it should be adjusted depending on the characteristics of your data. So if you see more rapid changes in the baseline that you need to remove, then you should increase that cutoff frequency of the high pass filter to get rid of that stuff. But it really is just dependent on what your data looks like, because what you'd want in the end is something that's more or less flat, centered around zero, and not a lot of deviations away from zero.
That change in 100, will that change the frequency the amplitude of events?
So yeah, so we'll get to that. So this is just the baseline file for right now, but we'll also apply the same high pass filter to the event file. It will distort the data in a way that may be undesirable. And anytime you filter the data, you could start impacting the shape of the events. I think though, because you're throwing out mostly low frequency information, and if the events are sharp in their shape, they shouldn't be impacted that greatly. But so one of the important things with your data is, review what's happening with every processing step. So if you think that your amplitudes are being affected by any kind of filtering, high pass in this case, it's good to really zoom in on the individual events and look at their amplitude both before and after filtering. So sometimes it can be hard to interpret just looking across the entire run, but if you just say pick out 4 or 5 events, zoom way in, look at their amplitude, and then see how that's changing with the effect of the filter, that's probably the best way to do it. That's not particularly scientific, but I think it's a good way to really understand what's happening when you introduce these different processing steps. Okay. So the after you remove the baseline of the background, it's centered around zero. Now we calculate the standard deviation of the points in the background, and that's what's up the cursor here. That's what's indicated here. So this 1.7 times 10 to the minus 11. And then you want to pick some multiplier. So when you find an event, you want it to be separated enough from the background. So the assumption is that the background in your event run will look the same as the background in just the background file. It doesn't contain events. If that's the case, you calculate the standard deviation and then you multiply it by some number, 3 times or 5 times. And that's what establishes the amplitude threshold. So then when you open the event file that contains peaks, the peaks have to exceed that value to be considered a valid event. So this is, actually quite simple, but I think this is still what's been done, typically to find events in the data. Right. So then the next step is you open up your event file.
The 3 times standard deviation, or 5 times standard deviation, that is just basically a product of analytical chemistry, right? If you have an idea of limits of detection, limits of, right, where you establish that. So this is not some exotic concept. That's basically, with 5 standard deviations, you have, the confidence that what you're detecting. So, it's 3 standard deviations already 99.
Yeah, right.
The 5 is really like 99.999. So that's pure analytical chemistry. don't think it's just
Yeah, they're not like magic numbers or something. That's where it comes from. Well, and I think, there's a lot of these things are debatable depending on what your experimental data looks like. So if you have an experiment where you have amplitude, events that are huge, their amplitudes are way beyond the baseline noise. You could probably be even more conservative, set an even more, you could use like 7 times the standard deviation. Then that way you make sure that you're only really, you're capturing events and there's no chance at all that you're capturing noise. But I think a more likely scenario is that the amplitude of your events will be smaller and you may have to be, more liberal than say 3 times. You may only be able to go 2 times or 1 time to find the events, to actually pick them out. But then that becomes questionable as, am I actually seeing now noise that I'm calling an event? So there's all of these trade-offs. And a lot of times too, you'll see here, well, my events are just way too small to measure. Is there something that I can do with my device or my experiment parameters to increase the amplitude of the events? Because really, as our analysis pipeline is established now, that amplitude is the most critical piece of information about the events, because that's what tells us if it's an event or not.
How fast are you collecting?
So I think right now we're sampling at 250 kilohertz. So, but that's a good point, because I'll talk about this a little bit later. We do start looking at the shape of the events. So we make measurements of the width, beyond the amplitude, but we'd like to use more shape information. So 250 kilohertz may not be sufficient to get enough points per peak,
Right.
particularly if they're moving through, and there's a lot of faster. So there's a lot of interest in trying to up the throughput of these devices.
Underestimated. Most of them underestimated. It's not fast enough.
Right.
but that's a bandwidth, right? So
It's,
acquisition
yeah,
time and bandwidth, I'm reminded always that those are 2 different things.
yeah. So I would call bandwidth something that is applicable to the transimpedance amplifier. So I think of bandwidth more in the analog domain. So the TIA has to have sufficient bandwidth to be able to pass through the frequency content that's high enough that gives you that more, more of the shape information. So like if you have very fast transition, sharp edges, the bandwidth of the TIA has to be high enough to allow you to represent those edges accurately. So this is all analog. You could plug the thing into an oscilloscope and see this happening. The sampling rate is when you take that analog signal and move it into the digital domain, meaning you're turning it into numbers that go into your computer. So that's related because that's how often you pick a point off of this continuous signal. So that also has to be sufficiently fast. But they're 2 related but different concepts. One is more analog, one is more sampling digital, but they do work together.
To answer your question, any data that has not enough data points, meaning not more than 3, it's not being considered a true event.
Right, so if we think of that event as, half of a sinusoidal peak. So in order to determine do you have enough points, you have to go 2 times the frequency, Nyquist limit.
Mhm.
But I think you might be way above the 2 times frequency, but that would be something to calculate to see.
Yeah, so if the events look like sinusoids, that would work. But I think as the events become sharper, so like
Right.
those, then you need to have higher bandwidth and more samples to
Right.
represent that. So, but yeah, we're way above what the Nyquist limit should be on these things. Okay, so this is now the event file before any processing has been done. And you can see with this particular experiment, something happened in the middle of it where the baseline just shifted down for some reason and then came back up. So this is something you probably want to deal with in the analysis. Maybe you can just ignore this portion, but I can show you what happens. So we
Yeah. Turn off the lights, yeah.
well, yeah, I don't know. This is more like no, I get what you're saying that happens, but this looks like almost like something it's like the whole overall open pore current shifted down. Like, the pore got smaller for a short period of time and then bigger again. So the resistance changed. I don't know. Anyway, So this is the raw data, but then you perform the same analysis steps as with the background file. So the first thing you do is you correct the baseline. So you get everything centered around 0. But this shows one of the downsides of the high pass filter. I think this is kind of what you were indicating before. So like Colin explained yesterday, the high pass filter is essentially like a differentiator on your data. So when you have these transitions that you see here and here, if you think about mathematically, applying a derivative to that, you get this result. You get these big spikes, which is problematic. So the ones that are positive going aren't so bad because you just ignore them. We only look at negative deviations to find events, but this is a problem here because these could be misinterpreted as events when they're clearly not. They're just an artifact of the high pass filter. So we've talked about this and I've made some progress, but I need to get this implemented. I mean, there are other techniques for baseline removal that don't involve a frequency domain filter like a high pass filter. I mean, the simplest thing to do is just thinking about like fitting a polynomial to the run and just subtracting that out, just a purely, sort of linear time domain technique. You could either just remove the baseline or maybe the slope or start introducing higher order terms of the polynomial to capture more of these transitions. What Dr. Hall does in his lab, because his analysis is actually done more in real time, is he takes a small chunk of data, I don't remember what it is, maybe a second, and fits the baseline just to that. And then I like his approach because his amplitude threshold then it is dependent on the noise or the the baseline in that particular chunk of data that you're working. Where everything we're doing here is all very global unless you just on your own select a subset of data to work with.
You could look at what's the peak width of that whatever noise,
Mm-hmm.
and you could impose boundaries. I am not
Yeah.
doing anything
Right. Yeah, and we do that. I mean, we'll see that next. We put dwell time windows on, so yeah, that may just drop out. But yeah, it's concerning that it doesn't really look like a real peak, but it's still, yeah. But yeah, I mean, I've dabbled with a few other things. There are these wavelet transforms that you can use, which sort of they start to decompose the signal, kind of like a Fourier transform, but into these more like, I won't get into it, but you can basically decompose the signal down into baseline components and the higher, shorter time duration components and throw some of them out. Some seem to work better than others, but I think I'd like to implement more of like what Dr. Hall has in his software, because that seems to be an effective way to go. All right, so what we did here, though, just to simplify things, is we just picked a subset of the data that doesn't contain this. So from here on out, we're just analyzing from 0 to 40 seconds, and that's defined here. And then what this shows is when you use that amplitude threshold, that's what's indicated in these lines here. The negative one is the one that we're using. This shows you now where the events actually are. So the peaks that cross that threshold are considered the events. And just to review, this number comes from the background file, which in this case, it was a completely different experiment that was run. And this was the number, the standard deviation of that data multiplied by 5 to give you that threshold. And that's what's shown here. So I wanted to make a comment. So I think, theoretically, your background should be a different file. You should be able to collect your background, establish the noise level of the system, and then pick an amplitude threshold based on that. But I think some students in the lab have found that when they add their analyte, that the overall background noise goes up. So collecting a run without the analyte in it isn't really a fair indication of the noise of your experiment. So this is debatable, but if you run an experiment where you add your analyte and you have some time before the first peaks start to occur, you could take that initial, I'll call it quiet time, and use that as your background, if that's more representative of the actual noise in your experiment versus a separate run. But knowing that it's a quiet time is debatable, because if you pick up a period of time that has events in it and you don't remove those events, and you calculate the standard deviation across that, including the events, that number is going to be artificially high, which will then make your amplitude threshold even larger, meaning that you may no longer find the smaller duration or smaller amplitude events in the data. That makes sense.
background should be from the run of the when you have an analyte, but you need to establish the threshold, you just You just choose the fragment of the baseline where there are no events.
Yeah. Yeah, that's a good summary. So is that what people in the lab are doing now pretty consistently? Or does it vary from experiment to experiment? Okay, because see I think that's changed in the last year. I think a year ago, the argument was you should have a separate run where you know for sure there's not an analyte in.
You have to have a separate run to see that it's quiet, that you don't see events because of some artificial noise
Mhm.
or some kind of error related to the system. So the baseline is absolutely necessary to get a confidence, right?
True,
Thing is
yeah,
that-
right.
But for my opinion, my humble opinion, when we do analytical chemistry, right, measurements, if I do HPLC, I don't establish the noise level from the previous run when I had the
Mhm.
baseline. I establish this from the run when I run
Right.
a light.
Yeah, no, that makes perfect sense. Yeah,
See, I see the threshold line here, but if we wanna assign uh, the peak height, the amplitude, for example, if it's 100 picoampere that shows on the data file after we analyze it, does the 100 picomolar, it start from the threshold line all the way down, or does it start when it actually the very top or when it upper, when we actually see the drop, like the beginning of the drop? Or is it like from the threshold line?
oh, okay, so yeah, let's see if
Yeah, so it's,
here's a zoomed in individual peak. So when the measurements are reported in the file, I think there's two things that are reported. So we report the time of the first point of the peak that crosses the threshold, but we also report the amplitude of the greatest deviation from zero. So that should give us this point down here. So not the point that crosses the threshold, but the greatest deviation from zero, because that's really indicative of the actual amplitude. Is that what you meant?
so it's from 0 to the tip.
From 0 to the tip of that, yeah.
Not from threshold to the tip.
No,
Oh.
it's 0, yeah, from 0 to the tip. So that's the measurement of the amplitude.
Thank you.
All right, so you establish a threshold, then the program automatically finds the events. But in addition to just crossing an amplitude threshold, there are other rules that are put in place to limit the peaks that are considered valid events. So the other things that we look at are the dwell time, which is the width of the peak. And then in experiments where you have two pores, we expect there to be two spikes, two events for everything that translocates through the nanochannel. So you get a spike from the first pore, some period of time, and then a spike at the second pore. So you can also use that time, the time of flight or the time to traverse the flight tube between the two pores is another criteria for establishing if this is a valid event or not. So those are the numbers that you put in here. But the point is then, you get this Excel spreadsheet that gets generated from the data that contains these variables that have been measured from the events. So I'll move on here. So what we're dealing with these dual pore devices are 5 variables that represent the peak pair. So we have, like I just explained, the time of flight, which is the time between the two peaks. And then for each peak, we have its amplitude and its dwell time. So we have 5 variables that we can work with when we're trying to, in a lot of cases, trying to identify certain things like certain DNA bases based on these signatures. So the first thing to do when you collect data like this is to look at the histograms of these 5 variables. So let's just talk, we can just use it as an example. This is RNMP data. So there's four RNMP types that we're looking for, A, U, C, and G in the data. So you would hope that if you create histograms of, and imagine that you ran an experiment that had all 4 mixed together and were coming through the system, and you didn't know what was coming through at any given time, but you would expect there to be 4 distributions because you have 4 different things. And that's what you want to look for. So you'd want to look at histograms of time of flight and then of the amplitudes and the dwell time. And that's what's shown down here. So these are histograms of time of flight. for the A's, U's, C's, and G's. And what's really nice about this is that you can see the time of flights are quite different. They don't overlap with each other. I mean, there's a little bit of overlap on the tails. But, this is, so from my, engineering perspective, this is one of the things that I think is really important about the devices from Sopra Lab, is that these two pores that are in series give you this time of flight information. And from what I've seen in the data, this is the best way to discriminate different things. So this is the variable that gives you the best discrimination for RNMPs, DNMPs, and viral vector capsids and all of these things. And this is not available if you only have a device with a single pore in it. So I think this is one of the really, really fundamental improvements with the technology that's been developed in the Sopra Lab. That and the fact that you can replicate these things in plastic, so you can make lots of them cheaply. But I think in terms of like the signals that you get out, this is really critically important. So this is a very basic look at the data. Again, this is just looking at time of flight, and you're seeing that it's identifying the 4 different R types pretty well, but there's still some overlap on the tails, like here.
I don't think you increase the length of the channel. Nanochannel. Is that right?
And that's a good question. So I know that some good, what happens when you adjust the length of the flight tube between the pores?
Now you see the pore interactions of the animal with the so principle you should have a longer time of flight.
So effectively, if there was no interaction, all of those things would come through at the same time, same time.
Not necessarily. There is because they do still have different electrophoretic mobility, right? What you're looking is it's basically a current mobility. Current mobility is whatever the electrophoretic mobility of each of the molecule and the EOF, right?
Yeah, so the structures not are exactly the same. So the structures are different.
Yeah, so they do have different.
So even through solutions without interaction with the solid surface, there still will have different mobilities.
Yes.
So what I'm thinking on the x-axis, if you increase the time of flight by increasing the channel length, then you might get a
Better
better
separation.
resolution, better
Yeah.
separation.
So
Yeah.
yeah, so now that I'm looking at this, actually, I think panel A is with a 5 micrometer tube and B is with a 10. And this is just showing A's and C's. So if you look at the A's and C's here, I think they are better separated. The tails don't overlap. And this is in a paper that I think Sunguk at LSU,
It's tricky because then it's becoming spread. So there's a fine balance. You are absolutely right saying that. But yes,
Yeah, the only thing to pay attention here, the longer the flight tube is better to separate, it will give you a higher identification accuracy, but it should be long enough to contain 2 molecules at the same time within the flight tube. Then
Right.
yeah, it should be short enough so that only one nucleotide or one molecule should appear in the flight tube at any given time. The longer it is, then it's gonna be a higher probability that 2 molecules can
Right,
enter
you
together.
get peak overlap and
Yeah,
something when a runner runs around a track, the fast one eventually
Right.
catches up. to the
right,
slow.
exactly,
Yeah.
yeah.
Yeah, that's gonna be the.
Yeah.
that might be.
That's-
Yeah, we have done a lot with these 2 examples that we had in our lab with the 5 micron and 10 micron and we actually saw like the separation was much more efficient. But as Vishat said, besides the possible movement of more molecules at that time, we also are changing the electric field. We're changing the resistance, so we need to pay attention to all those factors because then we can affect the electric field and consequently decrease the driving forces on the molecules on that channels as well.
Right, yeah.
So there's kind of a limit, right, that we can go further, but then at some point we start to lose some other factors as well as capture rate, for example.
Right.
so it's an art of compromise.
Yeah, the trade-off, that's
BB's not very good.
We do this on solution chemistry all the time, right?
Yeah,
Have some levels. You can make the, when we get overlapping peak, one of the things we do just lengthen the gradient process, how we scan the organic,
Mhm.
right? But at some point, if you do too much, then yes, artifacts begin detailing, confronting,
Mhm.
such as stuff.
Yeah. Okay, any other questions? All right, so, time of flight is very important, but we do have these additional measurements that we've made. Again, the amplitudes of the two peaks and the widths of the two peaks. So the question is, can we use that information to help us get even better separation between these, R and MPs? So one technique that we've used is principal component analysis, which I mean is a fairly standard data processing technique, but the whole idea of this is that, we have 5 original variables and we're using the principal component transformation to move them into a new space where we still have 5 variables, but now they're not the same. They're actually linear combinations of the original variables. So we have 5 original variables and 5 principal components. Principal components consist of, maybe a little bit of the first variable, which would be like dwell time, maybe a lot more of the second variable, which would be time of flight, and so on. But the whole point of this is that the interesting information in the data is how the variance. So basically how these populations look different from each other, because we're trying to separate out these different R and MP types. So what happens with this mathematical transform is that most of the variance is captured in the first principal component, then the next most in the second. So now we can just concentrate our effort on looking at the first maybe 2 or 3 principal components instead of all 5. And part of this makes visualization easier, that you can make plots in two dimensions and 3 dimensions. But the whole idea of this is that we want to get better separation between these things that we're looking at, these 4 different DNMP types, or sorry, RNMP types in this data. Okay, so this is a result of the principal component analysis, and we ran it 2 ways. So we started with just 3 variables or 3 features of the data, just basically anything that was time related. So time of flight and then the 2 dwell times. And what happens when you convert those variables to the principal component space and you plot them in 3 dimensions is you get data that looks like this. So all of the A's, so in this case, we knew what these things were. There were 4 separate experiments run. One only had A's, the next one only had U's, and so on. So we know what these things are. And every point in space represents a peak pair that was collected in an experimental run. So the ones that are green are the A's, the red ones are U's, and so on. And what you look for then is, so you've got all these points in space, and you can tell that they're sort of grouping together into these 4 constellations. And then we used a technique called k-means clustering to automatically partition all of the points into 4 clusters. So one thing you have to do with the k-means is you have to tell it how many clusters you think you have. And we know we have 4, so this is a good way of parsing out the data. So when we do this, these points are assigned to 4 clusters, and what's indicated in this table below is how well each of the R and MP types is assigned to a cluster. So like all of the A's end up in cluster 1. So there's 71 A's, they're all in cluster 1, so those are all identified correctly. But the U's, only 63 of the 71 end up in cluster 4, and 8 of them are misidentified and assigned to cluster 2. And then the C's and the G's are all okay. So the ones that are misidentified are the U's and the C's. Those are these here, and you can see that those clusters are overlapping with each other. So some of them are being misidentified. But what's interesting about this is now if you change the PCA analysis to go from 3 variables or 3 features up to all 5 that we have, so adding in the amplitudes, so it's the normalized amplitudes of the peaks from the 2 pores, your results look like this. So we have 5 principal components, but we're only plotting out the first 3 here. And now you can see there's very good separation in these constellations of points. So when you go through and do these, the look at every peak pair where it ends up, they're all identified properly. So this is great. We thought, okay, this is perfect. We can use PCA with all 5 variables on lots of experimental data, because every experiment that's run is generating these 5 variables, whether it's RNMPs or DNMPs or capsids or whatever you're trying to measure, you get the same kind of data. So we thought, okay, we'll just use PCA and that'll be fine. But what we found then with different data, so this is data that Kershed has collected and analyzed, and other people in the lab are continuing that work. Now we're looking at DNMPs instead of RNMPs. All right, so we performed the same operation. We just looked at the time variables to start with, the time of flight, the two dwell times. And then just the two-dimensional, three-dimensional PCA, and we got pretty good results, around 98%. So we thought, okay, well, let's do the same thing and add in the amplitude information like we did before. And we did that, and things didn't get better. It stayed around 98. And actually, when you look at three principal components, it gets worse. And if you look at all five, it drops way down. So we thought, well, okay, 98% is not bad, but we would like to get 100% identification accuracy. So we moved on from principal component analysis to machine learning. So I won't get into this in too much depth unless people have questions, but basically what we built was a neural network classifier. So the idea is that our neural network looks something like this. The hidden internal layer is a little smaller than this, but we have 5 input nodes that correspond to the 5 variables that we're measuring. Again, that would be amplitude, the first peak, second peak, dwell time of the first peak, second peak, and time of flight. And then we have 4 labels on the output, which would be the different DNMP types. So what we did is we trained this model on known data. So we said, okay, for this, this is a peak pair. We know this is, say, for example, an A, like we see in green here. So these 5 variables correspond to an A. And then, so we had this training set that was labeled, meaning that we told the model this is what it was, and the model learned on that. So it adjusted all of the internal parameters to give you a good mapping between the 5 variables that were measured and the label. And that's what this graph down here indicates. So, over the 1,000 training epochs that we used, I mean, we didn't even need that many because it hit 100% earlier than that. But with the training data that we gave it, which I won't get into this, but it's internally partitioned into training and validation data, you get 100% accuracy. The fact that both of these lines go up to 1 is what's important about this. So that means that for the training data, that every peak pair that was well, the model was able to map the peak pair, the 5 variables of the peak pair to its appropriate label. But what's nice about this technique is that after the model is trained and you see good performance like this, you can give it data that it's never seen before. So you can give it a new peak pair that it hasn't seen, and it'll tell you what it thinks it is. So what we did to confirm that this was working correctly is that we removed some peak pairs from the training set. So we pulled out 4 A's, 4 G's, 4 C's, and 4 T's. So the model wasn't trained on these peak pairs. The model never seen these before. But when you run the 5 variables associated with these peak pairs through, it tells you what it thinks it is. So like for the first 3 peak pairs, it said, okay, I think these are A's because it produces a very high probability number for A's. And these numbers that come out are actually coming out of these 4 nodes here. So you see those 4 nodes, that gives you these 4 columns. And because you see a high number here, that says, okay, the model thinks this is an A. And because we know what these are, because these are things that we pulled out from the training data, we can confirm, yes, those are A's. And then we knew that the next 3 were G's, and yes, it's predicting those are G's because it has a number very close to 1, or in this case, 1 for G's. Same for C's, same for T's. So this is great because now we're seeing that it trained adequately on the training data. It's working well on this test data. But what's cool then is we can use this for like an automated base caller. So if we run an experiment now where we don't know what the peak pairs are, they're either some kind of a mixture, or we're trying to do sequencing and we don't know what base is coming in which order. We just take the 5 variables from the measurement, give it to the trained model, and then just use the label that it provides. So we hope it's accurate. I mean, what we're moving to now is taking a known strand of DNA, so having a known sequence, doing all the stuff requiring the enzyme to isolate it and then clip it a base at a time and making the measurements. But if we know what the DNA sequence should be, we can compare it with what the trained model is telling us and make sure that they correspond to each other, hopefully very accurately. So this is what we're working with. And this seems to work well. We've applied this also to the like the viral vector project for looking at empty and full capsids. The difference there is that this is simplified, where instead of having 4 nodes on the output, we just have 2 for empty or full. But these models are all completely customizable. I just wrote the code using a Jupyter notebook. This is all just Python using Keras and TensorFlow, which is all pretty standard stuff. All right, I think that was about it. particular. So any comments or questions? I'm curious if people are analyzing data, even if it's not RPS data, using similar techniques, or if you have suggestions for what you think might work well. So don't be shy.
Do you? Thank you.
Yeah.
I would imagine that small changes in your device and you know, you would see different dwell times depending on the forces involved there. And also like peak amplitudes. So when you develop a training set, are these from all like numerous different experimental setups?
That's an excellent question. It should be, and that's what we need to go towards. So yeah, if you expect there to be variability from device to device, it's going to impact the signals that we're collecting, then yes. I think we need to collect a big enough, our training set needs to be built from basically the extents of the ranges of things that are going to change in the device. So, if you think that you're going to have pores that vary between this size and this size, we need to capture data from all of those in order to have a representative training set, because that's the problem. If the machine learning model learns on training data that's not generally applicable to new data that's coming in, and it may not be if the device is changing, it's not going to work well. So you need to make sure your training set is big enough to capture all those variations you're going to see in the device or the experiment, or anything like that that's changing. So I think right now everything we've done has usually been built off of I think usually, like in this case, there's 4 different devices that are used to collect the 4 different DNMP types. So that captures some of the variance, but I think it's probably better to be more systematic and make sure you're getting the full range that you expect to see.
Whether some data from RNA-based, DNA-based peptides altogether, will the system be able to find?
Oh, wow.
Apples to apples.
I mean,
You know what your assay is for. The assay is not for peptides, right? The assay is chemical peptides. Then the algorithm will be developed for max millions, right? You guys did peptides. Then the algorithm will be developed for the peptides, fingerprinting.
yeah, but I get your question. I mean, you could, as long as you train the model on whatever you expect to see in your mixture, it should say. But so this isn't something that I mentioned, but I should bring up. One of the other things that we're talking about is not just using 5 variables, but using many more variables, or I think ideally using the actual samples that represent the peak. So like, if I go back to this, if it takes, 5 time samples to represent the shape of the peak, or 10 time samples, or 50 time samples, we use all of those time samples as the input variables to the machine learning model. So now we're not trying to collapse this down to just one peak amplitude value or one width value. We actually are encoding the shape of the event by using those time samples. And, this goes back to the whole bandwidth sampling rate issue. If we increase the bandwidth and sampling rate, we'll have even more points. So I think in an experiment where you've got, this should help in experiments where the peaks look very similar to each other, where the differences that we're interested in may be lost from collapsing down to just a couple of variables like we're doing now. But I think it might also help when things look very different from each other too, because, like a peptide's not going to look like a DNA base coming through. And if that shape information is much, much different, and you do need to be able to identify both in the same experiment, that might help too. Yeah.
Because when you're saying training the model, right, it's not like training a dog that takes time. But could you make it's short, I mean,
It's quick, yeah.
calculations.
Yeah, I mean, it's pretty fast, mostly because we're only dealing with 5 variables per peak pair, and the model is really pretty simple. There's like,
Could you run events for each device that sends normally depending on how it's called, run the calibration? So could you run for example, here you have 4 different analytes.
Mhm.
So for each device, could you run automated training process with just injecting the
Yeah, you could.
analytes
train it and then use this model for this specific? Is
Yeah,
that
you-
realistic or it's just too involved?
Well, I mean, I don't know, I guess, you'd have to run basically like 4 experiments per device before you start running your real experiment. I mean, to me, that doesn't seem like that big of a deal, but I don't know, I mean, are the devices
You could do mixture, just like into that, right?
well,
And then she got those 4 distributions.
except to generate the training data, you have to know what the peak pair is. So you have to know, so I think because if you have a mixture, there'll be, so say you just mix A's and U's. Some are gonna be A's and some are gonna be U's, but you can't really assign a label to the peak pair when they're mixed together because they're coming through just stochastically. So I think you still have to have each one run in isolation first. So you have the label applied to it. But yeah, you could do that. You could build a training set for every individual device. And then when the model is trained, new data coming from that device would just use the training set from that device.
Can we do the isolation you talked about with PCA? Split it into 2, and then using parameters that we collect from PCA to neural network, to machine learning. If you don't, let's say, if you don't know how A and U look like at all, but we know that we have a mixture of A and U. Can we use both PCA and neural network to classify them?
Yeah, so actually in a case like that, PCA might be more effective because what you'll end up with is, sorry.
Yes.
So we know what these points are, but you don't have to have labels assigned. So you could perform the PCA and all the peak pairs are going to end up at some spot in this space. And you'll naturally see them clustering into, if you just had 2 bases, you should see 2 clusters start to form. So yeah, you could differentiate things that way without having labels assigned to them. And in fact, they sometimes call PCA unsupervised machine learning because it's unsupervised, meaning you can do this with data that doesn't have labels associated with it. Where what I'm showing here with the neural network, with the classifier, that's supervised machine learning because you have to have labels assigned to each piece of input data. Otherwise, the model doesn't know that it's making mistakes and it won't know how to improve or change the values inside the model to give you better performance on the next iterative cycle through.
Yeah.
That's a good point.
Yeah, I think we can go even after splitting into 2 clusters. We can go back from clusters to raw data, and then based on the cluster that it assigned, we can go back to raw data and then split that raw data into 2, saying that okay, this is the cluster A and this is the cluster B, which means this is A and this is U.
Right. Yeah, that would be a good approach. And
So we train the data using selected like for specific species. Yes.
right.
So it's not likely to happen, but just in case this thing we are using a new sample, but we will have a contaminant. Is let's say we are increasing DNA piece and we will have this more peptide. So will system will automatically remove that as an outlier, whatever that we get those data which we get for that contaminant.
No. So what you'll see, so for every peak pair that you give the trained model, you're going to get out 4 numbers like this. So it's going to tell you how much it thinks that peak pair looks like a G versus a T versus a C versus an A. So the hope would be that these numbers would be low, but it's going to tell you probably what it thinks it looks most like. So it gets to be a little tricky in a case like that because if the peak pair that's being generated from the contaminant looks enough like something that it thinks it is, it's gonna tell you that's what it thinks it looks like.
So the only thing that you can judge is time of flight, right? There's no other variable that you could train the model that to fit in.
So this is trained on 5 variables: time of flight, peak 1 amplitude, peak 2 amplitude, peak 1 dwell time, and peak 2 dwell time. And I think the rationale for why this performs better than the PCA that has all 5 variables is that this model is capturing nonlinear relationships between them. So I won't get into this, but like this internal layer is something called ReLU, a rectified linear unit. So it's taking the 5 input variables and combining them together in some complicated nonlinear way to give you the best mapping to the label, where PCA was just linear operations. So I think that's why you see a performance improvement when you do it this way.
But in our case, we have the dual ports, and technically they should be pretty close inside, but we saw a small asymmetry between the ports sometimes. So I was wondering like why it's important to have port 1 and port 2 measurements, and how exactly if they calculate an average or if they really know the distinction, because even between the experiments, if you don't standardize what the direction that you're using related to the device that you're using, you could have port 1, port 2, and then you flip your device to port 2, port 1.
Okay.
So I was wondering if you have considered this somehow in your models or if it's important or not.
So it's a good question, and it's come up before, and I probably should just go back and run it and see what happens if you just average the two numbers. So, if you use average amplitude, so if you average port 1 and port 2 amplitude and average dwell time and see if there's any performance difference or not. Yeah, I mean, if the pores are identical, then no, the second pore the measurement won't give you any additional interesting information. But I don't know. I mean, if there's something that happens in the experiment that would cause the signal, like the dwell time from port 2 to be different between bases, I don't know. I mean, if there's some complicated thing that's going on that we can't picture, it might be valuable to have that in there to capture that information. I don't know. But yeah, I think that's something that I need to try just to see, because I think my philosophy is use the simplest model that you can to give you the results that you need. So if we can drop this down to 3 variables instead of 5, and it's still adequate, that's probably what we should use.
So have to be single nucleotides or can it be a sequence?
These are all single. So are you like if we had chunks, like we had 3 bases or 5 bases?
Whatever sequence.
Mm-hmm.
And probably that goes through the port.
We have worked with, like, for example, since small homopolymers for, like, let's say 30 nucleotides, or heteropolymers for long molecules of single-stranded, double-stranded as well, or even like 4K bases or more. And it goes to the port.
It does go through.
Yeah, and depending on the size, it can go either as a coil or it can be stretched out and go as a strand straight away.
Again, signal events that sequence the single or double strand.
Yes.
But I think if you have, what are you saying, like a 10 of them, 10 bases in a row,
Signatures.
right. And you've got to, you basically have to train the model on every possible signature you expect to see. So if you have different orders of those 10s, you have to train it, and you have to train it on a sufficient number.
This is,
Yeah.
it's all true, but this is application specific
Mhm.
because Sheila's project, that's the whole point, to discriminate 30 versus 50 versus 70, because those are the signatures of certain information, right? Versus single nucleotide, it's for specifically sequencing project, right?
Right.
Where you will, based on stochastic, you will work with single molecule, either of RNA or single molecule of DNA. But that's determined at the input.
Yeah, one other thing I wanted to mention. So, we build the training set based on running experiments where each thing is in isolation, so we know what it is. And then I think the temptation is with the sequencing, then you jump to, taking a sequence of DNA and then, doing the full sequence, and then trying to compare it with like some sort of bioinformatics tools like aligners to a known sequence. But I think it can be valuable to take some intermediate steps. Like, it'd be kind of interesting if you, train your machine learning model and then you run a new experiment where you just have, say, G's and E. Well, you could have all 4, but one of them should have a much higher percentage. So, you have 4 times as many U's as the other 3. Collect that data, you'll have all these peak pairs. You don't know what they are. Let the machine learning model that's been trained tell you what it thinks it is. and make sure that the percentage of calls that you see matches what you know that it is. So, you should see, 4 times as many U's being reported as, and if that's not the case, then there's some problem somewhere. And I think it's easier to identify problems at that level than trying to understand why your sequencing doesn't align with the known sequence,
It's a great idea.
you know. And then, then you can take other, then like Adam Hall suggested, like if we're going to actually use a strand of DNA, pick a piece of the strand that doesn't have an equal balance of the 4 bases. Same kind of idea. If there's a lot more of one particular thing, run that through and make sure you get a lot more of that thing in the
Well,
calls.
That is true, but now you're introducing the sequencing.
Yeah, it's the enzyme and the digestion and all that stuff.
And A and T's are equally fast,
Mhm.
right? So they should arrive, right? There could be some slowdown. And we know
Right.
that the lambda exo is sequence specific, the rate of digestion, right? So no, this is good, but I like your idea with playing with.
Well, it's right. I mean, it's simple, but at least for me, it would give me a lot more confidence that the machine learning model is doing what you think it's doing. So, I mean, I get it. There's the temptation to jump to the end.
I'm
But-
gonna ask it. Right, this is a great idea to test it.
Yeah, and now that more people in the lab are learning how to run the machine learning on their own data, so that, yeah, good thing to try out. Okay,
Thank you so much, Matt. for- So that's officially closes the workshop.